Welcome to the Mutations video. We're going to go through this worksheet, which I've also added a codon chart to so that you can see on the same page, so I'm not going back and forth, but you should have this codon chart available to you so that you can use this to figure out which amino acids should be coming in the right spot depending on their codons, which again are up at the top: first base, second base, and third base. So let's get started. We're going to see the gene mutations, different kinds of gene mutations, and how they affect the creation of a protein. So let's start with a point mutation. A point mutation affects one single base, meaning one single spot on a big, huge piece of DNA is affected by this mutation. And a substitution is exactly what it sounds like. It's replacing one base with another base, one letter, A, T, G, and C with another. So let's start with an original piece of DNA. You can see here TAC, GCA, TGG, ATT. And again, they're kind of separated out into threes because of our mRNA codons. It's going to make it a little bit easier for us to do some transcription and translation. So the original mRNA we should read, and again, always read AUG at the beginning. Easy way to see if you're doing it right. Our next three are GCA, so just because of base pairing rules, we have CG and U. TGG is going to pair up with ACC, where are my college basketball fans? And AAT is going to pair up with UUA. So the amino acid chain, again, we need our codon chart to figure this out, but the amino acid chain again is going to be made from our mRNA. So our codons are located on the mRNA. So AUG, again, A is right here in the codon chart. G is the last one. That gives us methionine, and it also says start next to it. That's your way of checking to double check and see if you did it right. So methionine, we're going to abbreviate MET, typically just the first three letters. I'm going to put a dash here to show that there is a bond between amino acids to create this protein, and that is called a peptide bond. And then we're going to move on to the next one, CGU. C, G. So the first one up here would be for the U, but it doesn't matter because they're all arginine. So ARG. Which, if you want a really bad joke, what's a pirate's favorite amino acid? Arginine. Arginine. I know, it's hilarious. Moving on to ATC. A, C, C, doesn't matter. They're all threonine. And UUA, A is the third one here, and that is leucine. LEU. Alright, so we've gone from our original DNA, we've transcribed it into RNA, and then we've translated into a sequence of amino acids, which, given tens of thousands of these, or maybe hundreds of thousands of these, is going to give us a protein, if made correctly. So now we're going to hit our DNA with a mutation, and we're going to replace, right here, we're going to replace this C with a T. And this is something that does happen, and this is a way that DNA gets mutated. It simply changes one base with another base, nucleotide with another nucleotide, but the bases are the most important part. And again, this can be the effect of some kind of radiation, UV, X-ray, maybe a chemical. There's lots of different things that cause mutations. So we're going to say we replace this C with a G, or with a T. It's a substitution. Okay, we're bringing one base in for another. So after the substitution, we still have AUG, but now instead of CGU, we're going to have CAU. Everything else is still the same. So here we have our DNA, our mRNA after the substitution. Again, instead of CGU, we have CAU. Everything else is the same as it was before. So now our amino acid sequence after the substitution, again, we still have MET, because the AUG is still the same. We already know that these two are the same. Threonine and leucine are still going to exist there, but Now since we have a mutation, we have a new amino acid that's going to come in, which is going to change the structure of our protein, which most likely will change its function, or it won't function at all, which could be a big deal for the organism that needs this protein. So go ahead and use your amino acid, or your codon chart, and figure out what CAU is going to code for in terms of amino acids. All right, moving on to silent mutation. You can see here we've done substitution. We replaced the base. And a silent mutation is going to be a little bit different. You'll see how that works. Our mRNA, again, here's our sets of 3. Easiest to read them that way. We still have AUG. Now we have GCT, which is going to give us CGA. Still have ACC, and still have UUA. So, we go to our amino acid sequence after the substitution. Again, AUG we already know. CGA is our silent mutation. I'm going to leave that blank. ACC we know is threonine. UUA we know is leucine. So let's take a look at this one, or why don't you take a minute, pause the video, and see if you can figure that one out. Okay, when we take a look at CGA in our codon chart, I'm going to go back a page so it's a little easier to see. CGA in our codon chart. Oops, sorry, CGA in our codon chart. It's going to code for arginine, pirate's favorite amino acid. Alright, so here you can see how the mutation, even though it changed the sequence of DNA, and of course changed our sequence of RNA, it did not change our sequence of amino acids. Oh, what's up with this thing? It did not change our sequence of amino acids. We still have, as we did in our original up here, we still have arginine. Okay? So now you can see why it's called a silent mutation. We still have the same amino acid sequence. I'm going to use little a to abbreviate. Amino acid sequence means we have the same protein. So even though we've had a mutation take place on the DNA, which obviously changes the RNA, we did not change the amino acid sequence. And you can tell this can happen quite a few times. If you look at the codon chart, there's quite a few times where multiple sets of bases all code for the exact same thing. So good in a way, but also possibly could be bad in a way. But that's how that works. Alright, so we're going to move on to the next set of mutations. Still gene mutations, but these are called frame shift mutations. And a frame shift mutation, you'll see, makes a big difference. Larger difference than the last one. So we still have, let's go back to our original. Reading in threes. Alright, our amino acids chain is still going to be the same. Methionine, arginine, threonine, and leucine. But now our mutation happens. We're going to have an insertion where we put in a T where it used to read TAC. TAC has now been interrupted by adding in a T. We've inserted a T. So now our mRNA after the sequence is going to be AUA. Still keeping in line with threes. We have to keep the 3 code going. We have GCG. We have UAC. CUU. And when we get to that T, we know to make an A, but we don't have the rest of that code. Alright? So let's take a look at the amino acid sequence after insertion. AUA is actually going to code for isoleucine, which is not our start codon, it's not our start amino acid, but if the ribosome doesn't recognize it and doesn't know any better, it's just going to keep going. GCG is going to code for alanine. UAC is going to code for, UAC is going to code for tyrosine, and CUU is going to code for leucine. So you can see we have something that just happened to be the same, but the rest of our amino acid sequences messed up. If we had more after that, they would probably all also be messed up. So what you notice when that insertion takes place, all the letters get bumped down by one, which typically is going to mess up all of the amino acid sequence, except, for example, every once in a while it will leave something the same. It's possible. But for the most part, that amino acid sequence is going to be pretty severely messed up. Next type of frame shift is going to be a deletion, so you'll see the opposite is going to take place. We're just going to use a different example, just an English sentence: the fat cat ate the rat. Alright? So let's delete one of those. Let's pretend each one of these letters are bases. No coincidence that these are all three-letter words. Okay, we're pretending this is all codon still, but just using a different type of example. Let's delete one of those letters from our sentence. But again, continuing, the ribosome doesn't know any better, so it's going to skip right to that, what it thinks is the sixth letter, a C. And it's going to keep reading in order. And it just so happens that we still have some English here, but if you're to read this sentence to someone, they're going to think you're speaking a different language. Again, when you go back to the concept of translation, you're kind of translating one language of mRNAs into another language, amino acids. So it's kind of the same concept here, but again, once you get a deletion, now everything bumps down one to the left, and again, the rest of those amino acids past that point are probably going to be messed up, which could have a huge impact. It's probably going to have a huge impact on the creation of that protein. So these frame shift mutations, overall, you can describe them this way. It bumps down the code. If you bump down the code by just one is all it takes, by one base, everything past that point is going to be affected. Now the thing to keep in mind for mutations, although they could change the protein, changing the protein doesn't mean it's necessarily bad. There are actually probably thousands and maybe even more than that mutations that have occurred that have actually helped with the evolution of organisms. So mutations doesn't mean bad, it just means different.